Alrighty guys, so we're going to be going over mitosis and what that entails. So in this process, we have several things occurring. The first thing we're going to do is look at the overall cell cycle, alright? Now people refer to mitosis as if it's all of the cell cycle, but really mitosis is just a small part. It's about 10% of the cell's life. The rest of the cell's life takes place in this G1, S, and G2 phase. Now, some cells don't perform mitosis, like brain cells. Now, what do? Well, they go into a process called the G0 phase, where they just kind of go around in a circle. They don't really ever do anything. They don't ever replicate, they don't ever duplicate, they just live their life forever, basically, until they get killed. So here we go. The first part of a cell's life is G1. Now you might be wondering what G1 means. Well, this G right here represents growth, and this subunit 1 represents the first stage of growth, okay? So in this part of the cell's life, the cell's going to grow from little cell to a bigger cell, alright? Then it's going to hit this S phase, which is the part of its life, it's kind of like the adolescence of the cell. It says, alright, we are going to undergo DNA replication. So you might be wondering to yourself, well, what's DNA replication? Well, inside of a cell's nucleus, right? So here's the cell and here's its nucleus. There's this stuff called chromatin, alright? And what the chromatin is it's simply the elongated form of chromosomes. So this is kind of what a chromosome looks like. And chromosomes are made up of, you guessed it, genes, which are made up of DNA. Okay? So what DNA replication does is it goes to these chromosomes and it says, hey, it's time for you to duplicate, and an enzyme comes along, and instead of having just one of these, we duplicate it and we have another one, okay? Now, once we form this next level of chromosome, we have these two bond at a organelle called the centromere. Okay. Once these two chromosomes bond at the centromere, it's considered a duplicated chromosome, and each one of these different parts of this are called sister chromatids. Okay, so after it's undergone DNA replication, we hit a growth 2 phase. So it's growing to here, it hits its S phase, it duplicates all of its DNA, and then we go through this next growth 2 phase where it gets to its final size. Now, if you think of each percentage as a year of its life, you're going to undergo about 100 years, right? But in reality, that 100 years is equivalent to about 24 hours, right? Actually, excuse me. Most cells only live a few hours. Now, most cells, some cells only live for a few hours, some cells live for a few days, so it all kind of depends. But right here, if every cell's kind of congruent, if that makes sense, they all live 100 years, but each year is different times. Once it reaches about 90 years old, depending on the cell, that's all different times, but once it hits about 90 years old, it gets a signal to duplicate, right? Now, it gets this signal for several reasons. The first reason is because the cell, it wants to be replaced, right? So in stomach cells, the cell gets destroyed, right? So it needs to be replaced, right? The second part of it is if it just needs to be made more of, right? So when you get sick, you need more white blood cells, so your body makes more. And the third type is if a cell grows too big, it's going to say, hey, you need to be cut in half. All right, so let's take a look at this smaller version, but very important part of the cell cycle in a cell's life. It undergoes a splitting. That's what mitosis is. The purpose of mitosis is overall to get this DNA into two different cells. What you're going to end up with at the end, after you have cytokinesis, which is the final part, cytokinesis, once you get to cytokinesis, you're going to have two identical daughter cells, okay? But we'll get to that in a bit. The first stage of mitosis is considered prophase. Now, what happens to prophase? Well, this sort of chromatin in the nucleus of a cell is going to be condensed into these chromosomes. All right? Now, what happens next? What happens after that? Well, this nuclear membrane right here, it's going to dissolve. Okay? So it's going to dissolve, and the centrioles, which are these little people right here, are going to move to opposite ends of the cell. And these little mitotic spindles, which we'll talk about in a minute, start forming off of these centromeres. Now, mitotic spindles are made up of microtubules. And what they do is they're basically like arms that reach out and retract, grab and move these chromosomes. Alright, so what happens in metaphase? We can kind of exclude this pro-metaphase part. What happens in this metaphase part is the chromosomes align on what we call the equatorial plane, which is going to be right along here. And what happens is these mitotic spindles reach out and they grab onto each one of these chromosomes, right? Depending on the cell, it's going to have different numbers of chromosomes. So what's going to happen is these mitotic spindles are going to reach out and grab these chromosomes at, you guessed it, their centromere. Okay, and what's going to happen in anaphase are these chromosomes are going to be ripped apart at the centromere, and these sister chromatids are going to be broken apart and separated into sister chromosomes, right? And these sister chromosomes are going to be repelled or pulled by these mitotic spindles to opposite ends of the cells. Okay, now what happens in telophase? Well, in telophase, it depends how the cell gets separated. First of all, the cytoplasm in the cell gets duplicated and divvied up, right? So you have cytoplasm on this side of the cell and cytoplasm on this side of the cell. Now, what happens after that? Well, this nuclear membrane, which was dissolved here, reforms around these new chromosomes. So now you have these two nuclei, all right? These nucleolus. Then, if you're a plant cell, you're going to have these little vesicles, right, that form in the middle of these two cells. And they're eventually going to form into this solid cell plate, and then you'll have two separate cells around that cell plate. In animals like us, you're going to have a cleavage furrow that forms, all right? And that's when in between these two cells, they kind of get bent in on themselves. And that causes them to rip apart in cytokinesis, all right? So basically in telophase, what's going to happen is either this cell plate is going to form or this cleavage manifold is going to form. Now, in cytokinesis, that's just the splitting apart of these two cells.